Hi, everyone. Welcome to week 2 in child development. I wanted to just do a quick video to highlight some of the important concepts in chapter 2. So you will find in the outline of things to do this week in your checklist, the to-do list, that you'll need to watch several videos. And that's because I want you to have the same experience that my students in person have had as much as possible. And so some of those videos are a little bit longer. There's one about epigenetics I really don't want you to miss. It's so interesting. And I've walked to my classroom several times this morning to start this video. And for some reason, my OneDrive, my Google Drive, none of them are bringing up my slides. So I'm going to do a screenshot when I get back to my office and you'll see it above this video, hopefully. But I loved this quote, and this is what I really wanted to visit about today, is just the interactive nature of our genes. I think sometimes when we talk about genetics, we have to simplify it a lot so that we can understand this very complicated process and complicated concept. So we simplify it down very, very much. But really, it's important to understand that our genes interact with each other, that environmental influences have a big impact on what our genes do and that expression of those genes. And so there are just the smallest ways that our genes can be influenced within the cell, but then also within our bodies. Think about things how we take into our bodies, our nutrition, and how it can have an impact upon the expression of genes, and then our environment outside of our bodies, the things that we breathe, the things that we experience, even stress, and how they can impact our genes and the expression of that DNA. So I'm going to show you a video that came with our course. It's called Blueprints for a Baby. And so hopefully this will work.
Why do babies resemble parents in one trait and not another? We begin to answer this question with a description of conception or fertilization. An ovum or female sex cell is expelled into the fallopian tube, where it is penetrated by a sperm or male sex cell to form the zygote. The zygote contains 46 chromosomes, which are rod-like structures organized as 22 pairs of autosomes and 1 pair of sex chromosomes. The zygote becomes the morula, a solid ball of cells. These cells divide in a process called mitosis. Each new cell has the same number of chromosomes as the original cell. This is true for all cells except the sperm and ova. The sperm and ova divide in a process called meiosis. Each resulting new cell has half the number of chromosomes as in the original cell. Each sperm has 22 autosomes and either an X or Y chromosome. Each ovum has 22 autosomes and an X chromosome. This reduction is necessary so that when a sperm penetrates an ovum, the zygote will have 46 chromosomes, which is the normal number for the human organism. The new organism will be female if the sperm with an X chromosome unites with the ovum. The new organism will be male if the sperm with a Y chromosome unites with the ovum. The zygote, either male or female, contains the genetic code for the new organism. The genetic code, also known as the genome, contains all the hereditary information needed to build, operate, and maintain an organism. The outcome depends on the environment which surrounds the genome. Thus, even at this early stage of development, genes and environment are interacting. In order to understand heredity, we need to study the structure of the human genome, which is made up of a chemical called deoxyribonucleic acid, or DNA. The DNA molecule has two sides, which wrap around each other. The sides are sugar and phosphate molecules, connected by rungs of nitrogen containing chemicals called bases. There are 4 different bases in DNA: adenine, A, thymine, T, cytosine, C, and guanine, G. The genome is divided into chromosomes, with many thousand genes on each chromosome. There is a total of 30,000 genes in each human cell. Each gene has a specific place on a chromosome. Genes which regulate blood type, for example, are on the same chromosome in the same position in all humans. Genes determine which cells will specialize into brain cells and what color our eyes and hair will be. These genetic codes ensure that we are alike as humans, but unique as individuals. Why do some children closely resemble their parents while others do not? When the ovum and the sperm unite, the chromosomes of 2 different people are combined. It is during meiosis that the particular combinations of genetic makeup are determined for the egg and sperm. These variations occur during stages of meiosis. One possibility is the random separation of chromosomes during the final cell division in meiosis, where egg and sperm are formed. There are as many as 8 million to tens of trillions of possible combinations for each new organism. It is highly unlikely that any 2 individuals will have the same genetic inheritance, except for monozygotic identical twins. In the case of monozygotic twins, after fertilization, the morula splits in 2, and 2 separate individuals with the same genetic inheritance are developed. Another possibility for differences is the crossover process. During division, sections of the chromosomes cross over, creating new genetic combinations in the daughter cells. How are traits, physical, intellectual, and temperament, inherited? Genes come in pairs for each trait. If the genes are identical for a given trait, they are said to be homozygous, represented by a pair of upper or lowercase letters, for example, AA or AA. If the genes are unlike for a given trait, they are said to be heterozygous, represented as AA, seen here with an upper and a lowercase letter. The same gene will have different forms. The pair that mark these forms are called alleles. A particular allele of a gene may be dominant. This means it masks the effect of any other allele of the same gene. When an allele of a gene is dominant, only this gene needs to be inherited from the parent for the trait to show up. Among dominant traits are brown hair, farsightedness, and normal color vision. When an allele of a gene is recessive, the child must inherit the recessive form of the gene from each parent for the trait to show up. Among recessive traits are blonde hair, baldness, and color blindness. Some traits may be codominant, such as blood type. An individual who inherits type A blood from one parent and type B blood from the other will have type AB blood, which is different from either type A or type B. Most human traits are polygenic. Several different genes are needed for the trait. Traits such as eye and hair color and texture are polygenic, which accounts for the large variations in eye color and hair color and texture. Some traits are multifactorial. There is a potential for an inherited trait which is influenced by environmental factors. For example, height is an inherited trait but is impacted by nutrition. Most babies are born normal. Only 6% of newborns have a genetic disorder or a chromosomal abnormality. It is not unusual for a zygote to have more or less than the normal 46 chromosomes. Usually, this occurs during meiosis, when the sperm or ovum wind up with one more or one less chromosome. Most of the zygotes with more or less than the normal 46 chromosomes will be miscarried. Yet there are exceptions where the child will be born with some distinctive characteristics. The most well-known is Down syndrome, or trisomy 21. The individual has 47 chromosomes, 3 number 21 chromosomes, or, in other words, one extra copy of chromosome number 21. The Down syndrome individuals resemble each other more than they look like family members. They have almond-shaped eyes, flat noses, small heads, and short limbs. Mental retardation ranges from mild to moderate. With appropriate and early intervention, many Down syndrome children learn to read and write. The risk for producing a child with Down syndrome increases with the rise in maternal age. At the maternal age of 25, the risk is 1 in 2,500. By the age of 40, it is 1 in 100. Other chromosomal abnormalities involve having one too many or too few X chromosomes. For example, Turner syndrome, in which girls have only one X chromosome, XO, and Klinefelter syndrome, in which boys have an extra X chromosome, XXY. Intelligence, height, or fertility may be affected. Fertility problems may be treated with hormones. Genetic disorders may be the result of recessive or dominant inheritance. While most inheritance is polygenic, many genetic disorders are the result of one pair of genes. 1,800 different dominant disorders have been identified. In this instance, the baby needs to inherit only the dominant allele or gene for the trait from either parent for it to show up. Huntington's disease, which affects the nervous system. Polydactyly, extra fingers and toes, and achondroplasia, dwarfism, are examples of dominant disorders. For dominant inheritance, if one parent has the trait and this parent has children with someone who does not have the trait, then there is a 50% possibility that each child born of this union is going to have the trait. This is true for each pregnancy. If one of the parents has 2 copies of the dominant allele, then all the children will inherit it. Many dominant disorders are fatal, and the affected individual is either miscarried or dies soon after birth. In the case of Huntington's, the disease does not manifest itself until later in life. Each individual who has the potential of developing the disease must decide whether to be tested or live with the uncertainty. In order for the recessive trait to be inherited, the individual must inherit an allele for the trait from each parent, referred to as the carrier. At least 700 recessive disorders have been identified. Examples are sickle cell anemia, Tay-Sachs, phenylketonuria, and cystic fibrosis. If one parent has the allele for the trait and she has children with someone who is also a carrier of the allele, chances of having the disease or being a carrier for each child born will be According to these percentages, 50% chance for the child to be a carrier, 25% chance that the child will inherit the disease, and 25% chance that the child will neither be a carrier nor have the disease. If one parent has the allele for the trait and has children with someone who does not have the allele, for each child there is a 50% chance the child could be a carrier and a 50% chance that he could not be. Sex-linked inheritance involves a gene for a recessive trait, which is located on the X chromosome. Most sex-linked traits are found in males. A male who inherits the X chromosome with the trait from his mother will have the trait because the smaller Y chromosome he inherits from his father does not have the normal gene for the trait to mask the effect. Examples of the most common sex-linked traits are Fragile X syndrome, the most common inherited form of mental retardation, hemophilia, blood clotting disorder, and red-green color blindness. A female who inherits an X chromosome with the recessive gene for a particular trait from her mother will not have the trait because the second X with the normal gene, which she inherits from her father, will protect her from exhibiting this trait. She will be a carrier. In rare instances, a female may inherit an X with the gene for the trait from both parents. She will then have the trait. There are many genetic defects found most frequently, but not exclusively, in specific ethnic groups or in small geographically isolated populations. Some of these are Tay-Sachs, an enzyme deficiency that results in brain degeneration, is found in Jews of Eastern European origin. Sickle cell anemia, hemoglobin defect in which red blood cells take on a sickle shape. This is found in people from some African countries and African Americans. Cystic fibrosis, a lung disorder found in Caucasian populations. For multifactorial inheritance, in defects such as clubfoot, cleft palate, diabetes, childhood depression, and schizophrenia, there is a genetic basis, but something in the environment triggers the appearance of the defect. The environment refers to conditions in the cells, the uterus during the prenatal development, or to conditions in which the child is raised. The diet and health of the mother during pregnancy have an impact on the health of the child later in life. Sometimes chronic diseases, such as cardiovascular disease, hypertension, diabetes, cancer, and obesity, have been related to problems in the uterine environment during prenatal development. Genetic information inherited from each parent makes a difference. Diabetes, asthma, autism, and Huntington's are more likely to appear depending on whether the trait is inherited from the mother or the father. Behavioral genetics is the study of the strength of inheritance and the environment in which it is expressed. Nature and nurture cannot be considered separately. They are interactive in human development. Even within the cells, genes are responding to hormones and growth factors which activate or turn on genes. The relative effects of heredity and the environment have been examined by methods which are not controlled, as in most scientific studies. This is so because scientists cannot ethically alter the genes or environment of humans. In the family studies method, the effect of heredity and environment on physical traits is noted. For example, it may be difficult to determine whether all members of a family are obese due to a genetic tendency to gain weight early or to the faulty diet eaten by the entire family. In the adoption studies method, intelligence was shown to have both a genetic and environmental component. In one study, adopted children had an IQ more like their biological parents than their adoptive parents. In other studies, children who were adopted into stimulating environments had higher IQs after this exposure. Twins have been studied in a variety of methods. Identical or monozygotic twins were more alike than fraternal or dizygotic twins. Identical twins separated at birth were found to have similar physical and physiological traits. In the co-twin control study, one identical twin serves as the control as the development of the other twin is studied. In the chorion control method, identical monozygotic twins who were monochorionic, developing in one sac in the same location in the uterus, were compared to identical monozygotic twins who were dichorionic, developing in two sacs in different locations in the uterus. The results of these studies indicate that monochorionic twins are more alike in IQ and personality traits than are dichorionic twins, indicating the influence of prenatal experiences on development. In studying the hereditability of human traits, the concept of reaction range is helpful. Hereditary traits appear within certain limits. For example, a child born with mental retardation will do better in a stimulating environment but will not blossom into a genius. Physical and mental traits are the result of the interaction of genetic material inherited from both mother and father. The genotype is the individual's genetic inheritance, including both the dominant and recessive genes. This underlying combination of genetic material is expressed in unique ways. This combination of the genotype directs the possibilities for the unique phenotype of the individual. It is all of her characteristics that appear, including the way she looks and her personality. These characteristics may also be altered by events which occur prenatally or throughout childhood. Events such as war, famine, and disease will influence physical traits, such as age of menstruation, blood pressure, weight, and height. Climate, medical care, and nutrition also may alter phenotype. However, there may be limits or a reaction range for these alterations. For example, second-generation immigrants to the United States are taller than their parents due to improved nutrition, but this gain levels off after several generations, indicating that genetic limits have been reached. Personality traits or temperament refer to mental and emotional characteristics which are a unique way of acting and reacting to experience. These ways of behaving have been observed to be present at birth. They may be modified to some degree by the parents or the society into which the child is born. Different societies prefer different types of behavior. In the United States, competitiveness and independence are encouraged. In Asian and Native American cultures, cooperation and interdependence are prized. These values influence the ways parents parent. Sandra Scar and other developmental psychologists have addressed the importance of parenting. They have postulated the gene-environment correlation, which is that parents respond to the genetically driven characteristics of their children. Passive correlation. Genes influence both a child's environment and heredity. Passive because the child does not control it. An athletic parent provides both a genetic tendency for the child to be athletic and also encourages athleticism in the child. Active correlation. Genes influence what the child seeks out in the environment. An artistic child chooses activities which coincide with artistic interests. Reactive correlation. Environment reacts differently to people with different genetic profiles. Parents who are not athletically or artistically inclined may make an effort to provide either athletic or artistic experiences for their child. Intelligence, particularly IQ, has been studied. In earlier studies, it was believed that children were born with an IQ similar to biological parents. If the environment was sufficiently stimulating, the child would do well on IQ tests. Conversely, if environmental stimulation was lacking, the child would do less well on IQ tests. In other words, heredity provides the potential, and the environment determines the fulfillment of that potential. More recent research on brain development indicates that the environment in which the child is raised influences the way the brain develops. To quote from Neurons to Neighborhoods, experience triggers brain growth and refines existing strategies. The same environment may be perceived differently by different individuals, depending on their unique genetic makeup. The shared environment affects each individual in a similar way, regardless of genetic endowment. The non-shared or unique environment is anything else which may affect one individual but not the other. Molecular genetics is based on the new technology in mapping the human genome. The complete nucleotide sequence of the human genome was finished in 2003. The results reveal humans have fewer genes than previously thought, 30,000 in each cell. It may one day be possible to locate the specific genes which determine specific behaviors. A gene causing susceptibility to autism is close to being identified. Thus far, attempts to cure genetic defects in humans have not been widely successful. Some gene therapy experiments are being tested for cystic fibrosis, AIDS, and immune disorders, among others. In the future, it may be possible to remove defective genes from the fertilized ovum before the individual is born. When couples seek genetic counseling, one or both may be a person who have offspring or a relative with a genetic disorder, be a member of a high-risk ethnic group, the woman may be of advanced maternal age, or have a history of infertility or spontaneous miscarriage. The counselor may be a social worker, medical specialist, or genetic specialist who will take a comprehensive family medical history. The counselor may recommend a series of screening tests for both partners, which can include DNA, blood, and urine analysis. If the couple is already pregnant, diagnostic tests may be ordered: amniocentesis, chorionic villus sampling, sonogram or ultrasound, alpha-fetoprotein blood test, embryoscopy. One day, gene therapy may be a possible alternative. These screening and diagnostic tests help determine the risks but cannot prescribe what the outcome will be. The counselor uses a non-directive approach. He or she explains the risks of a genetic disorder for each pregnancy. It is up to the parents to decide what steps to take. They may decide to go ahead with a pregnancy or choose to adopt or try assisted fertilization. It is their decision based on personal, religious, and ethical beliefs. The interaction of nature and nurture works in two directions. The environment impinges on the way the genetic potential of an individual will be realized. The genetic endowment of an individual influences the way in which the environment responds to that individual. The beginnings of a baby shows the complexity of early development in the physical, intellectual, and temperament characteristics of the individual.